So in this video, we're going to be looking at the introduction to the blood chapter. Now we're going to look at, throughout the chapter, it covers the introduction, talks about red blood cells, different blood types, white blood cells, platelets, and how we make blood, and also how we control bleeding. You can go through the definitions on your own and have a look at the function, so I'm not going to spend much time there, but I want to spend a bit more time on a few of the key things. And that is when we start looking at blood, we can see that on average there's about 4 to 6 liters of blood in a typical person. And histologically, it's basically a liquid connective tissue. And of course, any tissue has both cells and an extracellular matrix. So let's have a look at those components. First of all, we have plasma. And plasma is the matrix of blood. And it's a clear yellow fluid. We'll talk more about what's in plasma, mainly water, electrolytes, antibodies, proteins, etc., etc. And then the other part are the formed elements. Now, most people would refer to them as the blood cells, but we have to keep in mind that one of the components is technically not a cell, and that's platelets. So really, we're looking at blood cells and fragments. So we have red blood cells, white blood cells, and also platelets. Now, when we look at the formed elements, there are 7 types of them. Number 1 are erythrocytes, which are red blood cells. The second one group are platelets, and as I said, technically they're not a cell, they're actually a fragment of a special cell called a megakaryocyte that we'll see down the road. Then we also have the leukocytes. Leuko, white, means like white cells. And there are a number of them, and they look different and they have different functions. Now, the 5 white blood cells fall into 2 categories, and the categories are granulocytes and agranulocytes. So just the name gives you an idea. So one has granules, visible granules, and the other one has no visible granules. And within each group, we can see we have the neutrophils, the basophils, the eosinophils, and the basophils. So if you notice, all 3 of these, their names end with phil. Then we have the agranulocytes, and the 2 types are the lymphocytes and the monocytes. Here's a quick diagram to show you some of the differences, and we'll just have a quick look. Here we have a monocyte. You can see a monocyte has you can see that it typically has this horseshoe-shaped nucleus. And one way you can start to tell these cells apart is whether they have granulars or not, or on the shape of the nuclei. Here's another type, which are the neutrophils. You can see it's a multi-lobed nucleus. That whole thing's a nucleus. Here's an eosinophil. You notice an eosinophil has quite a few staining granules, and you can see here's a very typical look. It's a bilobed nucleus that we often see. Another one is a small lymphocyte. You can see a very large predominant nucleus, and if you're lucky, you can actually see a little bit of clear cytoplasm on the outside. Now, often people confuse a small lymphocyte, let's go back here, a small lymphocyte with this cell down here, a basophil, because they both look very darkly staining. But a basophil, typically, you do not see a little clear band of cytoplasm within it. Here you can see the platelets. You can see the platelets are much smaller than the rest of them, and here is a typical red blood cell. Now, there's a term we should know, which is called a hematocrit. And that is basically if you centrifuge down blood and spin it for long enough, these components will separate, where it'll end up with plasma, a column of plasma, the red blood cells, and in between there's a little band that we're going to call the buffy coat. Buffy refers to a white area. And what do I find in the buffy coat? It's basically lymphocytes and platelets. And by spinning this down, you can figure out the percentage of each one of these components, which gives you some indication as to the health of the individual. Now, typically erythrocytes are the heaviest, and they settle first, and they make up somewhere between 37 and 52% of the total volume. The white platelets, the white blood cells and the platelets, they're the ones that we find in the buffy coat. They're typically about 1%. And then the plasma, which is this area up here, makes up the rest of the volume. It's somewhere about half to a bit more. So it's 47 to about 63%. And what do I find in there? I find water, I find proteins, nutrients, electrolytes, nitrogenous waste hormones, and gases. Now, when we're talking about plasma, we're really looking at the liquid portion of the blood. And we can also think about serum. Now, what's the difference? Serum is the remaining fluid when the blood clots and the solids are removed. So it's very similar to plasma, but the difference is that you remove something called fibrinogen, which you'll find at the end of the chapter, description of that. There are three major types of plasma proteins. We have the albumins. They're the smallest. They add to viscosity and osmolarity. They influence blood pressure and fluid balance. The next one are globulins. Globulins are antibodies, and in the immune system you'll find out more about antibodies. And there's different variations of that. And another major protein is fibrinogen. Fibrinogen is a precursor to a molecule that we need for clotting, which is called fibrin. And fibrin helps to form blood clots. And when we're looking at plasma proteins, many of them are made by the liver. The exception of that are the globulins. And the globulins are made by something called a plasma cell. Now I'm going to give you a heads up that a plasma cell is basically a B lymphocyte that has become activated to make antibodies. Are there non-protein components? Yes, there are. We have free amino acids, nitrogenous waste. We also have what you'd expect nutrients. We have vitamins, fats, glucose, cholesterol, phospholipids, minerals, dissolved CO2, O2, nitrogen, and not surprisingly, we also have electrolytes. And sodium is a significant cation that we find in there. Now, when we look at property of blood is much more viscous than water. It's about 4.5 to 5 times as viscous as water. And that's whole blood. If I look at plasma, it's about twice as viscous because of the proteins, etc. Osmolarity of the blood is very important, and of course, osmolarity is related to the dissolved particles within it. And if it's too high, the blood absorbs too much fluid, resulting in an increase in blood pressure. If it's too low, too much water, and this is really happening in a capillary, in terms of capillary exchange. If it's too low, too much water stays in the tissue, blood pressure drops, and we end up with edema. And just kind of like, the Goldilocks and the Three Bears, we don't want it too high, too low, we want it just right. So in the optimum osmolarity, we have the right percentage of sodium, protein, red blood cells, and the body works hard to maintain that balance. Just a couple points. Hypoproteinemia, this is where you have a lack of plasma proteins or deficiency. And this occurs in things such as extreme starvation, liver or kidney disease. Remember, many of the plasma proteins are made in the liver, and severe burns. And then we have some metabolic forms of it, where we see children with severe protein deficiencies. Often they're fed on cereals, which don't have complete amino acid profiles in the food. And they typically present with thin arms, thin legs. They're basically in starvation mode, yet you'll still see that distended abdomen, which is basically an ascites, a buildup of fluid in the abdominal cavity. Okay, we're going to leave it there as the introduction. Next time we're going to pick up the concept of hemopoiesis.